everyone. We're going to start our talk today, and I'll begin by introducing Dr. Harley. Hi everyone, I'm Ashley Moe, a Wisdom Officer, and I'm so excited to introduce our next speaker, Dr. Matthew Harley. Dr. Harley is an upper school biology teacher who was born and raised in Baltimore. He gained his PhD at Johns Hopkins University and completed a 2-year postdoc program in the plant pathology department at University of California, Davis. He has 2 first author publications to his name. Dr. Harley played basketball in college, and in his free time, he likes to play softball. He also loves traveling abroad and has visited China, Ukraine, Africa, Europe, and Australia. Today, Dr. Harley will be presenting on DNA sequencing and bioinformatics at Harker, with a highlight on Harker's new generation sequencer that will be used in a wide array of classes and for individual student research projects. Please help me give a warm welcome to Dr. Harley.
Hello everybody, thanks for coming. This originally was meant to be a nice casual presentation in one of the classrooms, but pretty quickly we saw the interest in the sequence program, and so we're here today in the auditorium. It's always interesting as a speaker to hear the bio, right? That bio I put into the Harker website 18 years ago when I started here, so I completely forgot what was coming. Okay, so indeed we are trying to call what we're up to here with this new next generation sequencer, we're trying to call it the sequencing program, because certainly that's what it's going to become. The incredible number of uses that we'll have for it, different classes, lots of opportunities for student research, and almost certainly lots of publications that are going to be coming out of Harker using this machine and, of course, the software for bioinformatics, much of which comes with the machine. Is actually on the machine, the software that we can use. So let me go ahead and get into it. Oops. And I must hit that button a while ago. Has that one been up already? So here is a picture of our machine. We have it upstairs. We had to adjust the room to make it air conditioned, to have it on its own power supply, to have a table that doesn't have vibrations because it is very sensitive, both while it's running and actually while it's not running. But wow, what it can do for us. And so a couple major things about it, and some of this that I'm going to be talking about, including the next few slides, is comparing it to more traditional sequencing methods, including some that we utilize here at Harker that we outsource and get some sequencing data. So one of the biggest things about next generation sequencing in general, and especially this NextSeq 2000 from Illumina, is how much data you can generate, that it's high throughput. As you can see up here, 360 gigabases you could generate. 3 billion bases can be sequenced in a single run. Versus more typical DNA sequencing using the Sanger sequencing method, it's 96 well plate, go at the next slide, about 800 bases per well in this 96 well plate, leads to about 75,000 bases only versus 360 billion. So it generates tons and tons of sequence data. And we went ahead, and of course all of this is thanks to very generous parents, the Pundy family. And as they showed their interest, we showed our interest back. We actually decided to buy a little higher end one that also does RNA sequencing. So what this will allow us to do is determine gene expression, whether genes are actually on or not. So it's not just looking at the sequence DNA, right? Everything about us, all of our different cells are different because different genes have been turned on, leading to different proteins. And so a ton of things that we can do with this. We're starting simple. We're starting with DNA sequencing this year, learning how to prep the DNA, making sure that we've got that done well, and then we'll move to RNA. Hopefully next year we'll get some projects with RNA, but a lot harder. RNA is much more sensitive, more fragile, so it'll be a new learning curve as we switch to RNA sequencing. Okay, oops, I lost the end donation. Okay, so as I mentioned, I want to compare a little bit, do a little bit of a lesson about sequencing, including, again, historic sequencing. But here we see the two mechanisms, that's not quite the right word, vehicles for sequencing. And I still use this in my biotech class. 96 wells, it can take 96 samples, actually 94, because the last two are controls. And again, about 800 bases that we can sequence per sample. Versus this flow cell. And it looks larger, it's not. This is it right here. And I tend to think of it being upright like this, but apparently with the label, I put my picture in upside down. And you can see, hopefully, oops, wrong button. This horseshoe-shaped structure here, that's the flow cell. And it has billions of nanowells. So as we put our DNA into this flow cell, they spread out and take up most all of these billions of nanowells. And that is a big part of how we can get so much, right, this high throughput, so much sequence data from this. Okay. And so again, a little bit of the history, and I'm not sure I'm hearing well. Is my mic's working? It's not breaking up? Okay, good. A little bit of the history of sequencing. So this is Sanger sequencing that is certainly decades old, 50 years or so. And what it does is it uses PCR amplification. So this is a technique where we can amplify a specific region of DNA. We can make more copies. And so shown here is just a very small, right, only 10 nucleotides. We'd be looking at DNA much longer than this. And not shown, but the PCR would have a DNA polymerase enzyme attach here to the 3' end of the DNA. And because G is the nucleotide, the base that complements with C that makes hydrogen bonds, it will attach. And then the polymerase moves down, and A comes in, hydrogen bonds, and then the chemical attachment covalent bond is made between them. So the G's put in A, C, T, G, A, G, C, T, because they're complementary. This is how it works in our body as well, in our cells. And so now this is doing it in a test tube, in a machine, but it's utilizing the same enzyme that we naturally make called DNA polymerase. And so this typical Sanger sequencing makes use of this. And what this is showing is that all of these nucleotides together, a small proportion of them cannot be extended from. So DNA stands for deoxyribonucleic acid. It has this 3' end that has an OH molecule, a hydroxyl group. It's needed to attach the next nucleotide in line. And so a small percentage of the G's that will be put into this reaction will not have that hydroxyl group that's necessary to keep that reaction going. So it ends that step, that extension. And so what we see then is that as PCR is working, most of the products will use a G that has that hydroxyl group, so it'll keep extending, keep extending, keep extending. But eventually, one of these nucleotides gets put in that cannot extend past that. And here in this diagram, it has them in different colors and it has little asterisks. A long time ago, when I was in high school, a long time ago, this was done with radioactivity, with a radioactive phosphate. And we would literally run them through gel electrophoresis. This separates DNA fragments based on their size. So the smallest DNA fragments can travel through the gel quickest. And then we can see. So the tube that we had some of these radioactive, missing a hydroxyl group, we call it dideoxy. those that ended in A, we can see how far they traveled and determine the size of that fragment, and therefore exactly where these A's are. Right? This one only has 2 nucleotides. It runs through very quick. These have 6 and 7 nucleotides, so they come in these positions. And then using these different tubes with the 4 different nucleotides, you could literally just walk up and say, okay, G, A, C, T, G, A, G, C, T, and we get the sequence. About 25 years ago or so, they switched from radioactivity to fluorescence. And so this color here is what's separating them, the wavelength of light that is emitted, and that the laser and the computer can read. And so now instead of 4 separate wells that each have a different radio-labeled, right, radioactive nucleotide, you can do it in a single well. And just use a computer. We put all of these together through a single lane in gel electrophoresis, and the computer will read. The first one that comes through had a blue fluorophore, it must be a G. A second one comes through, green fluorophore, it must be an A. So it's just literally going down the laser and reading off these, and we see that same sequence. And of course, complementary to the original sequence. Okay. The downside to this, especially now compared to this next generation sequencing, is that because these fragments are being edited, at some point we run out of extending DNA molecules, and the signal goes way down, right, as we use up all of these dideoxynucleotides that end sequencing. And so this manages for, again, maybe 800, 750, maybe up to 1,000 of strong signal, right, fluorescence, where you can read that sequence. Now, again, we are getting into a single sample for our next generation sequencer. We're talking about billions of nucleotides. And that's a little misleading perhaps. We're still using small fragments, but in a single well, we can now sequence tons and tons and tons of these fragments together. The computer can find where they overlap, put them into what we call a contiguous DNA sequence, that whole chromosome. It can match against databases to determine what species it is, right? What genes and chromosomes are there. Right? And of course, tons and tons of downstream applications. So now as we move into, and so I want to give you a real quick, I've been doing my biotech class for a while and still am. What we do is we go around to campus, we grab some leaves off different plants. The idea is we're going to try to genetically identify them. So we isolate the DNA from these different leaves. We do PCR amplification of one gene. It's a gene involved in photosynthesis, so all plants have it. We then have to take that gene and we insert it into a plasmid, a ring of DNA. Here it is. We have to put it into bacteria. We grow bacteria that take in this plasmid to make millions of exact clones of this DNA. Get it back out of the bacteria, and we send it out for sequencing, the outsourcing. But again, Sanger sequencing, old technology, it's actually quite cheap and easy to do. But we need at least 2 different PCR runs. So this is showing the primers, the small little sections of DNA that we put in that target where amplification happens, and then help start that amplification, right? Extending these DNA fragments for sequencing. And so we use 4 of these to get some redundancy in case 1 or 2 of them doesn't work. But each of these will do about 750, 800 nucleotides. We can put them together for a 1,200 nucleotide gene into the database, and we can match this gene to a specific plant. So there's pretty simple, cool, but it takes several weeks. We actually usually spend 4 or 5 weeks in this project, and a lot more steps than are now necessary. And again, not high throughput at all, 800 nucleotides or so per run in a 96 well dish. That's not that much output, it's just a single gene for these different plants. Now moving to next generation sequencing, and specifically the Illumina sequencing program. We're using DNA that we put together that looks like this. We have to make a library so we can take whole genomes, and we have successfully done this. We took 5 easy sources of DNA that we had, took a couple molds and algae, my colleague's DNA. We chop it up, fragment it into small little pieces, about 300 nucleotides. And then we add these adapters. This is for 2 reasons. This is for the primer, again, that will start the PCR process. It allows this section to be amplified and therefore sequenced. And it also has these 2 indices. These are little QR codes that the computer can then determine which sample it came from. So instead of 96 wells, separate samples into those 96 wells, we can combine a whole bunch of samples into that one flow cell, into a single tube, basically. And the computer, in reading these indices that get sequenced, can then separate them at the end and say, oh, these sequences came from Dr. Moore. These sequences came from the Pseudomonas fungus that we looked at, because we prepped them with these adapters, and more specifically the indices. So we end up, our whole genome fragmented into these small little bits, right, labeled with these adapters. And again, they can recognize which sample they came from, like QR codes, and then into the machine. And as they were teaching me this, they couldn't tell me some of the steps because it's proprietary information. And hopefully I do it justice here. But inside this, the flow cell, again, this little thing, this horseshoe-shaped flow cell, billions of separate little nanowells, each of which, or most of which, will get one of our DNA fragments. Again, the DNA insert, and then the adapters, the indices and the primers. They get attached by that one side, that one adapter, and then the amplification happens. So again, this is PCR based, but it's doing the sequencing as it goes. So to sequence, amplify and sequence the DNA insert, and then the index. So again, at the end, we can pull them apart and figure out which sample all the DNA came from. And then they do this cool thing, and this is the biochemistry they wouldn't tell me about. They add in the machine that has all these solutions which it releases sequentially, so this is all automatic. But it loops the DNA around to attach to the other adapter, cuts it off here, so it's basically inverted this upside down, and that does the PCR again. So for each DNA fragment, we're able to get reads from both ends. And it's called paired-end sequencing. And again, billions of these separate nanowells that each have a different fragment of our DNA that's been labeled. So again, we can backtrack at the end and see which samples these came from. So tons and tons of sequence data that we're getting off of this, from this process in this incredible machine and these solutions. Okay, so on to how we are using this. And again, Harker sequencing program. We certainly are using this for curriculum. I have already added it into my biotech class. So far, a pretty simple experiment. Certainly will get more complicated and fun as we move forward. And we are developing, Dr. Moore, my colleague from the summer, and he's been here part-time this year, including helping with the sequencing program, has developed a bioinformatics class for next year. So certainly is going to be utilizing this, doing multiple of these sequencing reactions, and then all of the downstream understanding about that sequence, right? Tons of different fields of biology that we can use to get this information for, whether it's evolution or development, including embryonic development, including gene expression, by turning genes on and off to different changes that are happening in the environment, right? Mutations, cancer, including diagnosis, right? Tons and tons of things that we can do with all of this information. And of course, importantly, actually before I mention that, and certainly it's going to be trickling down into our junior level, junior year biology classes as well, especially the AP class, especially the junior AP class where we have a summer portion and we have some extra time. But we'll be doing some of this soon. But definitely starting out in these two elective classes. Okay. And absolutely the most fun part of this is the huge possibilities for students doing independent research, potentially within the research class or simply on their own. Right? If they're signed up for a synopsis, their project gets accepted for synopsis, potentially me to be the mentor, certainly for this aspect of their project, to get DNA sequence or RNA sequence, to learn some aspect or something that has piqued their curiosity that they can learn about. And we certainly believe that we are the only high school to have one of these next generation sequencers to be able to do this. So the unlimited number of projects that our students can be doing. Fantastic. And now I want to take you into a couple of the projects that we are currently doing, or currently planning on doing. A simple picture here. Just yesterday, I had my biotech class try to obtain some bacterial colonies from their own skin, their own microbiome. We're growing these bacterial colonies, maybe some mold, maybe some yeast as well. Those are common on our surface. Getting colonies of them so we have lots of bacteria, lots of DNA. And now we can sequence this and see which species of bacteria are on you and the relative abundance of those species. Do a bunch of you know about microbiomes? They've certainly been in the news the last 5 years. A little bit. So the belief is that these microbiomes are certainly helpful to us, in part preventing nasty bacteria from taking hold. But it seems like they change very little over the long run. Everything we touch will get transfer of bacteria back and forth, but it tends to resort back to our original microbiome. The same species of bacteria and the same relative abundance. Most scientists believe that it's actually childbirth, passing through the vagina, where you first get this microbiome that you may keep your entire life with the same species of bacteria. And so what I tried to do last semester with this was I had students swab their hands, the backs of their hands, so it wasn't what they just touched. We tried to get the DNA out of that. They came back from winter break and did it again. So I wanted to see if it indeed had changed significantly. That experiment failed because we didn't get enough DNA just from swabs. So this semester, I've gone to this system. We're growing those bacteria to get millions of identical copies where we can isolate tons of DNA and get sequence information. The other benefit to doing it this way is that human cells can't grow on this particular medium. We don't want to get our students' cells, our students' DNA, right? Their privacy is important to us, their genetic privacy. This can't be on a database anywhere unless you choose to do 23andMe or one of those places. Ancestry.com, we don't want the Harker server, or worse yet, the cloud, to have your information. So this is a great way to filter out human DNA. One downside is that not all bacteria can grow on this medium. So we're not going to get the full microbiome, but we'll see a whole bunch of bacteria that are on your skin versus your classmate's skin. And again, in what relative amounts, abundance. And yes, maybe we'll do that second experiment swab them again in a couple of weeks and see if it's changed. Okay. Any questions on this? Of course, there's time for questions at the end. Okay. So this is the one simple project that we've already installed in biotech. And we definitely will get results from this now that we have tons of bacteria and tons of DNA that are growing. This is a picture. Again, this is my colleague, Dr. Moore. He's a huge hiker. He goes out into the hills around here. He collected these millipedes from the hills above San Luis Obispo, and they fluoresce. And we know about multiple fluorescing organisms, but they primarily use a single mechanism, luciferase. These millipedes do not. We don't know their fluorescence mechanism. So they also have not yet been sequenced, not yet been properly identified, and the mechanism of their fluorescence is unknown. So this should be a pretty simple, quick experiment to be able to find, sequence the whole thing and determine which gene is different from non-fluorescing millipedes that is probably causing this fluorescence. Okay. This, I added a couple pictures from the honors textbook, an older edition. This is actually showing some different species of cichlid, which is a common freshwater aquarium fish, but they naturally live in Lake Victoria in Africa. And what was discovered years ago, as more humans were coming to the shores of Lake Victoria, right, building up infrastructure, more fishing, more pollution, is that what was 500 different species of cichlid was dramatically reduced to about 200 different species. And yes, some might have gone extinct, but some of them started to hybridize with each other. With that pollution, both chemical pollution, they couldn't as well recognize each other's scent. Visual pollution, they couldn't see specific coloration, specific spots, and they started breeding with those that they hadn't before to see this hybridization of fewer species. There is a interesting hypothesis that this is happening here in the San Francisco Bay. We have a number of species of surf perch, both out in the ocean and in these different populations in the bay, where of course there's a bunch of pollution. So we are planning on going around, I didn't show a picture of the bay, but to different locations in the bay. We have a boat captain that has all these permits that can do this for us, collect surf perch, or at least little fin clippings. And we can be sequencing them, comparing them to each other, comparing them to the ones out in the ocean, and determine if indeed these fish are also hybridizing. And of course, downstream is figuring out why. Is it pollution? And of course, that can be a major way that we can try to help prevent pollution by showing the public some of the things that are happening because of us. Okay? And certainly one of the possibilities with this one is to be a long-term project, right? Every year, see if this is changing. There's possibility, Ms. Chetty is really excited about having some younger students start this, maybe in middle school. And then as they advance, can be doing more, right, different types of analysis with this DNA. And of course, the population genetics work that needs to be done, right, using this sequence. So hopefully we'll bring this program down into the middle school as well. And as they move through Harker, through the high school, they can keep adding extra research and understanding of different aspects of biology, including population genetics using these surf perch. And then this one is a question mark. And I put this one in there partly just in case I didn't emphasize the gene expression possibilities enough earlier in my talk. And hopefully I gave you some interest in that. that we can see which genes are active based on the presence of RNA. So by doing RNA sequencing with this machine, we can see how development is progressing in different organisms. We can see how they respond. We had a student last year that was trying to do this as his independent research. And of course, we didn't have the machine then, it couldn't be done. Certainly not by us and not cheaply. He was adding into these small worms, a model organism, adding in a chemical, and was looking for how their behavior would change. And he still did that just based on visual observations of behavior, but to actually see what genes were being turned off or on that determine that behavior is what would be really exciting. Okay? And again, this can be things like cancer diagnosis, not that we'd be diagnosing any human cancers here, again, for privacy reasons and other things, but looking at potentially cancer in model organisms, we'll be able to see which genes are maybe affecting that. Not just mutations, but whether they're upregulated or downregulated, being turned on more than they should, in the wrong cells than they should, or downregulated, right, turned down or turned off. So really cool stuff that we can do. And again, question mark. And yes, almost certainly we'll be thinking of some projects that we can be adding to classes, but a huge part of this question mark will be for the students to come up with different projects. Tons and tons of possibilities now. And then as I end here, I wanted to mention that we are already exploring different possible collaborations. Dr. Moore has a lot of contacts at Stanford. There are Stanford students that are already wanting to interact with us. And not just use our machine, but work with our students in doing their research. Okay, certainly some of these projects are big enough, important enough that the whole lab, right, the professors would want to be coordinating, collaborating with us. Maybe it's nonprofits that have interest in conservation, right? Redwood conservation that would be working with us, our DNA sequencers, our projects. So now we're talking about lots of extra help and of course, just working with research professionals, working with stakeholders, and of course, publications, and of course, especially if you think about nonprofits, maybe leading to action for conservation, for different things. So incredibly, incredibly cool things that we are now able to do. And again, thanks to the Pundy family for this fantastic, very generous donation. And that is indeed where I am ending. That went longer than I thought. As a teacher, I always tend to think of more things that I want to say as I go. I certainly still have time for any questions, or I make sure to add this, especially students, if you have ideas. And I won't steal your ideas, but if it's for a project and you're willing, then maybe it could become curriculum for one of these classes, or just to see potentially for your own future what project you might be able to do. Thank you very much.